All right, folks, what questions y'all have this morning? That's a lot. I don't know if I can answer all those at once. Give me a minute. So we want to explore some commonalities amongst the processes that we talked about, and we kind of deep dived into gastrulation and regulation events so far, movements and structures and terminology with frogs. I'll expand that a little bit, and you'll notice once you sort of know the terms and the basics of what's going to be moving and what we're going to call the places where it starts to move, we tend to keep using those same terms in other systems. So some of the other very common developmental model systems include zebrafish, Embryo, and they're a nice little aquatic organism that is easy to maintain in the lab. They make lots of eggs, and they're fairly good enough size. They're about a third the size of a xenophis egg, an embryo, but they're still large enough to do some manipulations with, to do some experimental embryology with. And so that they've caught on as another vertebrate model system, aquatic model system. And their early embryo starts out where you've got the egg with the large amount of yolk, and the cells, sort of the initial fertilization and the cellularization happens on just the surface of that, and the cleavage stage creates a dome of cells that is got some hollow space inside of it. So that's our blastula, and that's here. And you can see there's this is yolk, and that's the dome of several hundred cells. And then gastrulation happens at the edge. It starts on one edge, and that will be the future dorsal side. And it begins to ingress. And you can see that they're showing that here, that little gray loop there. Begins to ingress, and as it does, the cells continuing to divide will spread in all directions, down and around. I'm saying down because we generally draw it that way, but around the yolk to mostly include it entirely, not quite, but almost. And it also looks like it's going down and around, but it's not going around it equally. So on the head end and the tail end, you have movement, but on the sides, that movement sort of stops after a little bit, and you get majority of the embryo developing along the axis, along the future anterior-posterior axis, with just a thinner set of cells going around the sides. So the majority of the body is developing here, and you've got somites. You can see the segments right here. This one, the head is here, the developing eye is there, and the brain and everything is there, and this is the trunk. And then eventually down here will be the tail bud, and it'll grow a little tail, and then you have little free-swimming smolt larva, and then it'll develop on into the adult. And it's a pretty quick, about 90 days, so 3 months, and you've got yourself a new little fish. And the early stages are pretty quick. So you notice this is in days over here, this is in hours. So you're going through those same processes like cleavage and gastrulation and neurulation in about the same time span as xenopus, so you can do manipulations. The eggs are easy to inject and manipulate, and they can do experiments with it. Why this stupid thing doesn't work when you're in Zoom. So this just shows a little more detail about those cell movements. The first cell movements are actually the epithelial movements, and then the mesodermal cells involute and give rise to the mesodermal layer, and that starts on the dorsal side. So you do have sort of the lip. This is called the shield stage, where this sort of is moving down and kind of enclosing that. And your gastrulation movements are similar, except you don't have these cells. This is not cells. This is just yolk. It's just inside a membrane, but it's not like the endoderm cells were. So it's not cellularized. The only cells are in that layer around the top, kind of the main difference. So I kind of want to move on through the rest of these systems. We've got the basics with xenopus, and we'll see some of the things. But one of the things we can do with developing embryos is, of course, we want to understand what genes are involved in these processes. So as genes are discovered and we try to understand what they might be doing, one of our very first little tricks is to try to identify which cells in the embryo are actually transcribing that gene, producing the mRNA, and consequently making that protein, and see if that gives us some clue about where and what that gene might be actually doing. So here, for example, is one particular gene called snail, and looking in the early embryo at the shield stage, or what we would call in xenopus the initial gastrula stage, which would be like right around here when it's beginning to undergo gastrulation. You can see that some cells are expressing that gene, and they're stained dark. They're detected, and there's a colorimetric stain that's happening, and you can see that those cells are here. There's cells all the way around. This is the yolk. Down here, not staining very well. You can kind of faintly see it, but you notice it's kind of missing from this little zone right there. And that little zone is actually, there are cells there. Those are the involuting cells. So this tells you that, well, this gene, snail, and there's a reason it's named snail, we'll get into it later, but that particular gene is present in some of the cells at that area, which are mesoderm, but not in the involuting mesoderm at the dorsal lip of what would be the dorsal lip of the blastophore. And if we look later in development, after the notochord begins to form, it's not expressed in the notochord, but it's expressed in the cells immediately adjacent to the notochord. So, okay, that tells us something about that gene. So this type of experimentation is called in situ hybridization. Okay, you're looking in the cells, in situ, in place, and you're using some sort of a hybridization technique. In this case, you're using a probe made of nucleic acid, which could base pair with the RNA. So you might have a DNA probe that's base pairing with the RNA, or you might have an RNA probe that's base pairing with the RNA, right? Because nucleic acids base pair with each other. And that probe is labeled with a specific chemical that can be detected, and that's what's giving you the color. In this case, they just took a black and white picture. In reality, this is probably purple or blue, dark blue. Okay, you can also use immunohistochemistry or immunocytochemistry, either term. Immuno implies you're using some aspect of immunology. Well, it's really an antibody. You're using an antibody that would be specifically recognizing a protein. In this case, it would need to recognize the snail protein. Okay? And that antibody would be conjugated with a tag, either fluorescent or an enzyme, and develop a color. Okay? So again, you're doing the same thing. With in situ hybridization, you're looking for messenger RNA, and with immunocytochemistry, you're looking for the protein directly. So you're just looking for the gene expression, but sometimes you're going to be looking at the RNA, or you might be looking later at the protein. And with those two techniques, you get the same type of result. You can take the entire embryo, because they're small and they can be made permeable, so chemicals can get in and out of the cells, and you can stain them. Okay, and we'll either call it whole mount in situ hybridization or immunohistochemistry or immunocytochemistry. You'll stain them. Sometimes it's even just called immunostaining for short. And you can detect where the RNA or the protein is being expressed, which cells have it, which cells don't have it. And both pieces of information can be important. Okay? So here's another gene called no tail at the same exact equivalent stages. And it's kind of a nice, the reason I picked this one is because, okay, so no tail is expressed in exactly the converse group of cells from snail. So no tail is expressed in that group. It's also expressed in those cells, so it overlaps a bit. But it's definitely expressed in the notochord, but it's not expressed in the marginal cells next to the notochord. So this type of approach with embryo, with analyzing the gene expression in an embryo is very common. It's one of the first things that's done. My postdoc advisor used the acronym SYN. Okay? If you're a developmental biologist, the first action is SYN. Sequence the gene, do an in situ hybridization, okay? So sequence and in situ hybridization. And then do what's called a northern blot. And a northern blot can give you an actual abundance of the RNA. So it's a measure. First, you sequence the gene, then you look to see where it's expressed, and then you look to see how much of it's made. Okay? So he called it the SYN papers, because every time a new gene is discovered, those are the 3 experiments that are going to be in there. They're going to have the sequence of the gene, they're going to have an in situ picture of where it is in the embryo, and then they're going to have northern analysis of how much is being made. And then they might also have various manipulations, like could you have a mutant embryo or a mutant system and see is it expressed at the same place if you have this mutant. So this type of thing, both xenopus and zebrafish and chick and Drosophila fruit flies and sea urchins, they're all easy to do because they all develop out in a petri dish or on a piece of paper or in a test tube, and you can get access to the embryos and manipulate very easily.
So you said that no tail is expressed in all of the mesoderm cells that don't express snail?
Except, in the later embryo it is. But don't worry about where, I'm just giving that as an example of what we can do. So, chick, another vertebrate system used since the beginning of embryology. The identification of the initial germ layers was all done with chick. Okay, so mesoderm and ectoderm and endoderm and all that stuff was done using chick and staining it, because you can chip open the eggshell and cut a little window in the membrane, and you can actually manipulate the embryo in there, do things to it, and then just cover it over with the they would just use like an oiled paper. We would use saran wrap, okay, and it maintained the moisture inside the egg. It's fine. It'll just keep developing. You don't need the shell. You just need the shell to protect the embryo. If you provide some other mechanism for protection of the embryo, it will be fine. Okay, and so initial development takes place beginning on just one specific point in the yolk, on the yolk in the egg. Okay, so the actual oocyte was that whole yolk originally, right? And all of the stuff out here around it, what you would call the egg white, was placed in there around the egg, basically the telling envelope of the egg. And then the shell was placed on last, right? How does the sperm get through the egg-shell? It doesn't. So fertilization happens in the oviduct before the egg shell is actually produced, and then as the egg travels through the oviduct, it picks up out the egg white, the albumin, and it eventually picks up the shell, calcifies the outer layer. Well, all during that time, the embryo is already developing because fertilization happened before that. So there are several hours, about 20 hours, before the egg is ever laid by the chicken. Okay, and so during that time are the initial cleavage stages, so that when the egg is laid, you have essentially the early the blastula has been formed, and shortly after laying is when gastrulation then proceeds. And we were looking at embryos that were approximately this stage here at about 18 hours. That was our earliest one. This would have been at about the 48-hour to 72-hour stage embryo here. Oh, it's the same 50, so 48. I think they're drawing maybe a little past. And then we move into days. Of course, the organogenesis takes another 15 days or so, and chicks hatch somewhere around day 21, day 23, somewhere in that vicinity, and then take a couple of months to become a chicken. So here's the early, so the yolk would be there, they've just trimmed it away for clarity. And here also, they've trimmed away the yolk. You're looking at, say, this is probably around 48-hour embryo, and this is probably day 18 or so embryo. Just a little before hatching. So in that initial divisions of the embryo, what's kind of unusual, and this is also true with the seafish, the zebrafish, is the cleavages the cell divisions do not completely divide. So all of the cells actually maintain a cytoplasmic bridge or portal to the actual yolk. So the yolk itself, when you look at an egg, next time you make an omelet or something, the yolk was the cell, it was the ovum, right? And there's a membrane around it, the cell membrane bilayer. Well, it doesn't completely divide, right? So the outer edge of cells are sort of forming, but they don't form a complete division away from that. Eventually, the cells in the middle, like this one, would divide, and there would be a chunk of it that's now completely separated. Right? And that kind of continues. So you have an area here that is sort of open, and an area around it that's a little more opaque, and then an area around that that's a little more open again, and that represents incompletely cellularized blastoderm. And then this represents a bilayer blastoderm, the darker green, which would be like looking right through here. And then this area pellucida represents looking in the area that is essentially the developing blastocele. Okay? So eventually these cells will continue to divide and fill all the way across, and you'll have the blastocele form. That would be the blastula stage. And that's at about the time that the egg is laid, so right about 16 hours, 12 to 16 hours or so. These cell layers, as I was saying in class, I think I called it the hypoderm. So that layer is hypoderm, but each individual cell is a blast. So when you see the word blast at the end of something, just think it means cell. Cell in an early embryo, usually. Okay? Like from blastula. So epiblast, hypoblast are the two layers you have. And then when gastrulation begins, that is an initial delamination. So these cells are turning loose of the epiblast, and they're interacting with the hypoblast. And the first group of cells that move into there will replace the epiblast cells. Sorry, will replace the hypoblast cells with those delaminated epiblast cells, and that becomes the endoderm, the definitive endoderm. Okay, so they're showing you here that process. So the hypoblast cells are displaced as these delaminating cells, which would be called mesenchymal cells, take their place. Those cells are definitive endoderm. They eventually will displace that entire layer because everything's getting bigger at this time. The migrating cells are migrating both laterally from the involution area, which is causing a little groove to form. The anterior zone of that groove is our Henson's node. And really the only thing that makes it different morphologically from any other part of the groove is that the cells there go in sort of three directions. They come in and go sideways, they come in and go kind of at an angle, they come in and go straight towards the head, the anterior, okay? The cells at the Henson's node are the equivalent, well, kind of the entire street are equivalent to the blastopores, okay? Blastopore lip. Henson's node is kind of equivalent to the very first involution, the dorsal blastopore lip, right? So that continues, and that's visible at, say, that 18 to 20, 24-hour stages. And you can kind of see, okay, so it starts out, this would be that early 18-hour stage or so, maybe a little earlier. 18 hours, usually you have begun what's called regression of the node, and as it moves back posteriorly, the reason it's moving back posteriorly is not because it's actually moving, but because this is getting longer. So it looks like it's moving, but it is moving a little bit, okay? And as it's moving from its furthest anterior position, that's where notochord begins to form. And all the cells that move in ahead of it, so Henson's node would be here, and it would have been here before, all the cells that would have been here before. So as it's doing that, as it's moving back, the cells that are moving in become the axial mesoderm, okay, the cells right from the node, which give rise to the notochord.
How do you see that again, like the scales?
The cells that ingress at Henson's node and then continue to ingress as Henson's node moves more posterior become the notochord. So this red dot, as it's moving back, in its wake is left the notochord. Okay. Well, from its anterior position, kind of draw a line across there, you'll see that's there, that's the same spot, but that spot is right there. So where do these cells come from? The head fold starts to grow, but the notochord does not underlie that area. Okay, so that's a characteristic of that kind of separates some chordates from other chordates, right? So you have maybe the earliest chordates, the tunicates, and they're known as cephalochordates, right? Cephalo meaning head. The notochord continues into their head, okay? You have, I can't remember, the lancet, whatever that thing is, amphioxus, right? Then you have full-fledged chordates, euchordates like us, where the notochord never progresses into the anterior cephalic region. The notochord stops at the neck, okay? all this stuff is developed from not notochord. There's same genes are doing it, but those genes are just expressed in tissue that does not organize itself as a notochord. It's called prechordal plate. Prechordal plate. And it's technically endoderm. Okay? That's where it came from. Right, so as that continues to form, where the notochord was, you start to form somites on either side of the notochord, and the head, the induction of the head continues to develop. So basically, the embryo is elongating in two directions at once. The head's growing out this way, while the tail is growing back this way, and they're both growing simultaneously. And that's where you would be at say, close to the 33-hour embryo we were looking at, where you can sort of get that little hammerhead shark appearance of the neural tube developing up there. The what will become the atria, the heart, are starting to form, and so on. And then as it progresses a little later, you start to see the structures. You see neural plate down here, where Henson's node is just forming. The neural folds in the wake of that, the neural tube in the anterior. So it's a wave of events with chick, whereas with say zebrafish and frogs, it's more or less simultaneous. Almost the entire neural tube closes simultaneously, but in chickens it starts closing anteriorly in what would become the neck, and then continues posteriorly until it gets to the end. And even before the neural tube is completely closed, you already have developing, say, wing buds, and then leg buds, and so forth, starting to form. And you can see cross-section, the neural tube has been closed here. You've got somites on either side. There's a little notochord, a little knot right there. And you're starting to form hollow pockets in here amongst this lateral cells, which will be like the dorsal aorta and the stela. And body cavities starting to form. All right, how about our buddy the little mouse? Okay, so with a mammal, embryonic development is taking place in the uterus, and with the placental connections, and the observation of that is a lot trickier. You've got to essentially sacrifice the animal to access the embryonic development. And once you remove the embryo from its connection to the uterine lining, well, it's not going to last very long. So there's a limited amount of things you can do. You can remove eggs and fertilize them in vitro, and they will develop through the blastocyst stage because they're not normally connected to the uterine lining at that point anyway. But they won't go beyond that. They have to interact with the uterine wall in order for those cells to actually be specified. So in mammalian development, unlike what we had going on with, say, Xenopus, the cells, as they're forming the blastocyst, are not undergoing any sort of cellular communication. They're all equal until the time of implantation. And implantation into the uterine lining, it's actually the interaction with those uterine cells that are releasing signals, and those signals then trigger the differentiation of differences within the blastocyst. So the initial ball of cells has essentially a hollow ball. In one area, there's a larger aggregate of cells, and that aggregate is called the inner cell mass. Okay, the inner cell mass. It's a mass of cells inside the blastocyst. And the blastocyst for mouse, and even for humans at this stage, is probably around 128 cells. So a very few number of cells. That's only 2 to the what? 2 to the 4th, 5th, 8. Anyway, it's only been a few cell divisions. Not like in Venus where the blastula proper was after about 10 cell divisions. This hasn't been that long, and it's taken about 48 hours to get there, 48 to 50 hours to get there. For a human, not that long for a mouse, only a couple hours for a mouse. And so once it implants, then this inner cell mass and the outer layer of cells differentiate distinctly from each other. The outer layer of cells becomes what's called trophoblasts and invade the uterine wall, and the inner cell mass grows and becomes the embryo proper. But there's a lot of it that also turns into what's called extra embryonic cells, and those extra embryonic cells are what gives rise to the placenta. So the placenta develops from the embryo, right? One aspect of it does. So you have a lot of development that is all about forming the connection and not have anything to do with the actual embryo itself, with the component of the embryo. It's definitely necessary, but it doesn't, it's not going to be in the thing that gets born, okay? So
what did you say about the extra embryonic cells?
extra embryonic cells,
What did you say about them?
they develop from the at implantation to form the placenta. Yeah. So gastrulation takes place. In mice, this embryo is kind of cup-shaped. In humans, the embryo is essentially, instead of, you form this little cup, but instead of the embryo being cut, the embryo is essentially flat, like a chicken. So really, things are happening are the same, it's just sort of the topology point. So sometimes we say human embryogenesis resembles more like chicken embryogenesis because you have a flat embryo and you can see the primitive streak and it's sort of the same exact movements in a plane, but in a mouse, you have all those same things going on, just imagine it being made into a cup, right, or a basket, right, like a, probably rather than a cup, you would think of it as like an urn, some people will describe it, because it also has a top. The top is the extra embryonic stuff. So basically there's a line where all this stuff is growing into the uterine lining and this stuff is growing out of the uterine lining. And that cutoff is right about there. So in looking at mice, it gets a little tricky to try to draw. I've never found pictures that satisfactorily explain it to me and kind of have a good idea about it, but you basically have a group of cells that are the epiblast that are developing. They develop from that inner cell mass, and you have these cells on the outside. Those are called trophoblastic cells. Troph means what? Heterotroph, feet, right? So they're kind of called that because they're sort of the cells that interact and eventually participate in the formation of the placenta. There's the polar trophectoderm and the mural trophectoderm. Well, the polar trophectoderm is on the side where you have the inner cell mass. They are different from these because they're the side that's going to interact with the lining of the uterus. These cells are not uniformly able to interact. Okay, so it kind of bounces along until those cells make some contact. Those cells form your extra embryonic tissue, the placental cone, and go on to develop connections. The whole half of the embryo that's basically embedded, so where that constriction is basically everything that will be inside the endometrium. That's all extra embryonic tissue. It's mesoderm and ectoderm and endoderm. It's all three, but it's extra embryonic. So here you have kind of what we showed with the chicken. You've got one sort of level of cells, the blue ones, which are always confusing because where are they located in the embryo? Where does it look like they're located in this cup? On the outside of the cup or the inside of the cup? The inside. They're not. They're on the outside of the cup. The surface of your cup is contiguous with the outside of your cup, right? If you have a cup, right? it looks like maybe you should call that the endoderm because it's on the inside. Well, it's going to move to the outside when you turn your cup inside out, okay? So, saying cup is not a good thing because you think, well, cup, you can't bend a cup inside out. So think about a sock. Maybe a sock is a better example. When you turn your sock inside out, the part that would be touching your foot is going to be on the outside. And that is the future, the epiblast. It'll become the ectoderm, okay? And the outer layer, which is in contact with what would have been the yolk, is the endoderm. And as those two cells are triggered by signals coming from the endometrium, they begin to differentiate, and along one side, a group of cells starts the migration process. It looks just like a Henson's node, except it's not called Henson's node, it's called the node. And they start to ingress. And they move in between the blue layer and the yellow layer, and they displace the yellow layer, and they become the endoderm, and then they become the mesoderm. So that part works just the same, except instead of it happening like in the chicken, where you had these cells move into here, let's look at it as if we were looking at our cup, not from the side, but from the top, okay? So it's open. You've got cells here, and you've got cells here, and there's a layer between them, but there's not a space between them yet, okay? And so cells from here move into that in between, and as they start to move in there, they move out on either side. And if you look at the cup from the side view, really don't like that whiteboard. They moved in here, and they moved in here, and they're moving around the edge. Okay, so they're going around, they're going down. Down is that way. So they're going to go down, they're going to go around, right? They're going to go around here further here. So they're going to basically form kind of a wedge, like that red wedge that's there. So that red wedge, it becomes the mesoderm. Some of them become the endoderm, but then the rest of them become the mesoderm. So that's a better picture. Cutaway. So you have the original cells that move in, and then the secondary group of cells that move in, right? And the original cells that move in are neither the node or anything. They're the primitive street, but then at the cup tip, that's where you have the node. And that's where everything starts to take change. And everything from here forward becomes the head, and everything from here back becomes the tail. So where the node is, this from here up will be the head, and from here back will be the tail, trunk and tail. So this has flipped it, all right? So this is showing you that the ingression is happening on the right side of the picture. It's now over here on the left side of the picture, which is irrelevant. They just turned it. The red stuff is your, come back. The red stuff is your involuting mesoderm, right? And you got precordial stuff moving in first, and then you have the node, right? So everything here will be the head, and then everything that happens here will be the tail. And it continues that development. So here's the head developing, and you're looking at a sagittal section of it. And here's the somite starting to form, and it's coming on back here. Eventually, it gets to the point where inside this cup, you're kind of got the whole interior filled up. The initial little mouse is taking up kind of a central spot inside there, right? And so what's on the sides? It's just one or two cell layers thick, and that's what's represented here. Okay, that's the developing allantoic membrane. Okay, that's what in a mouse, that's or in a human, right? That's the sac of fluid that's going to be right around the developing embryo and fetus. So like when the water breaks, you're a pregnant woman and the water breaks, that's what's breaking the allantoic membrane. Well, the mouse is kind of inside out. His back is here, his belly is here. How do you get that so that the back is on the outside, the belly is on the inside? It undergoes what's called turning. And it basically just twists in there. And when that happens, the allantoic membrane is twisted around and you basically twist it off into a pocket. And the embryo is inside that pocket, and where the twist came together will form the umbilical cord. Okay? And then you continue to develop. So all this happens inside that blue layer. That entire blue layer was part of the embryo. So it's known as extra embryonic ectoderm. And then the red stuff is extra embryonic mesoderm, which gives rise to the blood vessels of the umbilical cord and the placenta, which is developing here. So mice are pretty hard to visualize without like watching some videos. So I'm trying to find some good videos of the mices. Fruit flies, everybody's favorite thing, right? Mm-hmm. Yeah, so fruit flies are cool and fast. Of course, invertebrates. A lot of different stuff going on here. Everything we've talked about so far has been invertebrates. It's had the chordate things. It's had a node cord, okay? Eventually a neural tube, so on and so forth. Well, what about invertebrates? So with invertebrates, we find that there is a lot more initial information about specification for anterior and posterior, dorsal and ventral going into the actual egg during oogenesis than there is for any vertebrates. Okay, xenopus and zebrafish have quite a bit, and sea urchins have quite a bit. Of course, sea urchins are also invertebrates, but they're like us. But you have maternally produced mRNAs that are transported into the oocyte, and you also have maternally produced proteins. These are produced in cells that are not the egg, but are associated with the egg, and they have a connection, a portal that they can transport material into the egg through. So the egg is absorbing these materials, and as they enter the egg, they are partitioned or localized to one end or the other based on microtubules that are in the egg. And so you have, when the egg is produced, already an established polarity with the presence of certain messenger RNAs and proteins. That's not entirely apparent just upon casual observation. This wasn't known until experimentation with things like in situ hybridizations and immunohistochemistry showed that, hey, this protein's just in that half. But as a consequence of early cell divisions, the material that's localized at one pole becomes now sublocalized only to the cells that form from that half of the embryo, or that very tip end of the embryo, or in the middle of the embryo, and so forth. So as the cells divide, each cell is essentially grabbing or being able to partition within its cytoplasm only certain amounts or only certain types of proteins and messenger RNAs. So there's an initial polarity that's established during oogenesis. Okay? The cleavage stage in many invertebrates is what's called syncytial cleavage. So you actually only have karyokinesis taking place, only nuclear divisions. And the entire group of developing nuclei are maintained within only one cell membrane, the original egg membrane. In fruit flies, that happens 13 times. You have 13 nuclear divisions, and on the 14th division, cells form. And at that stage, you have what's called cellular blastoderm. Right before that, you had syncytial blastoderm. So for 13 divisions, so 2 to the 13th, you can calculate how many nuclei are there. And then on tube the 14th, they form cells. And at that point, well, the cells at one end have gathered entirely, trapped an entire different set of RNAs than the cells at the other end. And some will be anterior, some will be posterior. This is, as it's drawn, this is the future anterior, this is the future posterior. And then gastrulation takes place. Now, in invertebrates like this, insect, gastrulation starts on what will become the future ventral side, the belly side of the little fly, okay? And those are shown cartooned here, again with kind of our same color scheme. Ectoderm or neuroectoderm is blue, and mesoderm is red, and endoderm is yellow. So the mesoderm forms on one side, and that side is the future ventral side, and it involutes and forms a tube, just like a neural tube, except it's on the ventral side, and it's not a neural tube. So in other words, it's exactly like it, but it's not it. The only part that looks the same is you have that involution, right? And the actual cellular movements are very, very similar to and the shape changes are very similar to the shape changes to get a neural tube, because there's one way to make cells form into a tube, basically. They can go boop like this, so one side gets wide, the other side gets skinny, and they all go boom like that and fold over. So that's what happens. Here's the cool thing. The same genes that cause the fruit fly cells to do that are the exact same genes that cause the frog's neural tube to form, and the chicken's neural tube to form, and your neural tube to form. So the genes are conserved. There's what we call a homology of process. There's a developmental set of controls that you use. You're just using them in a different spot in the embryo. And then once that happens, the neuroectoderm can form neural cells, and that process is very different from the way it is in chordates. So we won't make a comparison and contrast in there, except the process is very different, but the genes are the same. Okay? They're working in a similar way, but in a very different morphology. So with gastrulation, you have some differences there, but you have some homology of genes involved. This shows sort of a scanning EM picture. So looking at the ventral side versus at the early stage versus the later stage, and you have the initial groove, ventral furrow they call it, and then it closes, and then the embryo begins to undergo some expansion, but it's trapped inside of a membrane, just like frog eggs are. So if we look at the dorsal side, you can see that this furrow kind of turns the corner, and you can see there's the two sides kind of coming together. But it begins; these cells begin to push anterior and back that way posteriorly, and that's called extension. So the embryo undergoes an extension, and then it undergoes a retraction of those cells. And in the process, these cells, these are important cells, they get placed inside the embryo on the dorsal side of the embryo. So this is at full extension. This is the ventral side on the left and the dorsal side on the right. The anterior is at the top, and posterior is actually right there, because this is going to suck back around. Okay, so here it is at full extension, and here it is after it has retracted. And what do you notice about this embryo? They look like somites, but they aren't somites, because A, they're not made from mesoderm. Mesoderm is inside already. These are just external ridges created by the extension, and each one of these would be a segment. And if you think about an insect, what do you think about? Segments, right? Think about a worm or an invertebrate like that, think about segments. So this is where segmentation of the body pattern comes into play. All right, so we got to quit. It's good stuff, but we got to quit. So We'll try to finish that up next week, and then we'll get into what's really in your book. This is kind of covered in chapter 1, but they don't do it quite the way I'm doing it. So kind of reference that, but then we're going to get into some cell specification stuff next week. So I think that's chapter 3 in your book. So we'll kind of start matching up with what we got in the book.